Thank you very much, and I'd like to express my deep appreciation to the Environment and Health Fund for the invitation, and not just that, but for their extraordinary hospitality. So what I'd like to tell you about today, the main points of my talk, are that we believe there are such things as obesogens that may contribute to this burgeoning obesity epidemic, that at least some part of the action of obesogens has to do with reprogramming the fate of a family of stem cells in the body. And I'd like to show you our recent data suggesting that the effects of at least one type of obesogen exposure are permanent and heritable. So as you know, as you see, there is this worldwide epidemic of obesity. In the US, it's particularly bad. This number is now 35.5%, are what we call clinically obese. It's twice the worldwide average, and when I was in Korea a few years ago, they were very happy to say that it's 10 times the obesity rate in Korea. Many of us are overweight, and that number is estimated to grow very large. But I'd like to comment a bit about this measure that we use, BMI. These are slides which I stole from Lars. And these are two obese Swedish men from his PIVA study. These are MRI images, and you can see here in gray the fat. And this person has tremendous amount of fat under the skin, which is very unsightly, but probably not so unhealthy, where this person carries fat in the abdominal cavity, which, as you heard, is associated with much of the pathology of obesity. And these two men have the same body mass index. And here's another person with the same body mass index, and you can say many things about this person, our former governor, but obese is not one of them, yet the body mass index is the same. So we have three completely different phenotypes with the same body mass index. So we need to be careful how we apply this measure. And as you heard, something like 20% of obese people are metabolically healthy, and there may be as many as 40% of people who have a normal weight who are metabolically unhealthy. So this is a slide from the US Centers for Disease Control showing what's happened with the rates of obesity in the United States just since 1990. And as you can see, the prevalence of obesity has changed very dramatically. And there's something else that's also changed over the same time period, and I always offer the beverage of your choice to someone who will tell me what these numbers mean. Any guesses? Any bold students here? This number represents the number of health clubs adjusted for population, which has also doubled over the same time as obesity has doubled. So clearly, we're trying not to become obese. Here's the recent map for obesity in Europe, and as you can see, there are pockets of obesity in Europe, but it's not nearly as prevalent as in the US. Unfortunately, the Greeks are the most obese population in Europe, and I saw this t-shirt in the Plaka in Athens a few years ago, so in addition to their other issues, obesity is interfering with their ability to reproduce. Seriously, the problem with obesity, as you know, is that it's very expensive. It siphons a lot of money out of the healthcare system that we might better use to do other things. In Israel, you have very nice universal health care. In the United States, we don't. So if we didn't waste so much money on conditions associated with obesity, perhaps we could make sure that everyone is insured the way you have it. It costs something like $210 billion annually, which is a serious number even by Washington standards. This obesity, the costs largely derive from increases in metabolic syndrome and type 2 diabetes, cardiovascular disease, hypertension, stroke, cancer, days missed from work. You can make lots of associations. So how do we get fat? And we all here, I'm sure there's a few medical doctors in the room, and many medical doctors in the US say, well, you're fat because you eat too much and you need to exercise more. And obviously that's true, right? Because you can't get fat from breathing the air. You have to consume more calories than you expend. But at the same time, it's not so simple as just balancing your checkbook, right? Because if it was, no one would be obese. Because it's not a desirable thing to be. We try hard, we build all these health clubs, we exercise, we diet, we try hard not to be obese. So if it were so simple as just balancing the checkbook, there wouldn't be a problem. Here's an example of a person who eats more calories than he burns, a gigantic sumo wrestler. Are there other factors? And you've heard that there may be some other factors. We know that stress, associated perhaps with elevated glucocorticoid levels, has some role in obesity. And we know that not sleeping enough, in addition to other health problems, is also associated with obesity. There is this model that there were such things as thrifty genes that evolved in our hunter-gatherer days, and people who had the thrifty variants of the genes that we're talking about did a better job of storing calories that they consumed. And once that was a competitive advantage. If you stored more calories that you consumed than you passed on out. Perhaps that's not such an advantage today when calorie-dense food is highly available, and we hope that it never becomes an advantage again, because that'll mean civilization has collapsed. We know that there are such things as adeno-associated virus 36 is associated with obesity. We know that the kind of bacteria that live in your guts are associated with obesity. The ratio of Bacteroidetes to Firmicutes bacteria in your intestines are related to whether you're obese or not. And you can do a fecal transplant from an obese mouse to a lean mouse and make that lean mouse become obese, and vice versa. So there are many things that we don't understand that have something more to do than just calories in, calories out. And we know that there are changes, single nucleotide polymorphisms in a variety of genes that are all associated with obesity. What about prenatal nutrition and developmental origins of disease? We know from the studies originating at the University of Southampton that prenatal nutrition has an important outcome later in life, and that mothers who were undernourished produced babies who were small for gestational age, who were predisposed to obesity and diabetes later in life. We know from a huge number of studies that maternal smoking causes decreased birth weight and later obesity. As far as I'm aware, there's not a single study that goes against that. And there was a paper a few years ago, actually it was kind of a silly paper, by a woman named Paula Bailey Hamilton, who said that the reason she couldn't lose weight after she had her babies was because she was full of chemicals, and that if you followed her detoxification diet, you would get lean. And she showed that the obesity epidemic roughly correlates with what you heard, the increased use of a whole host of chemicals since World War II. Well, I just showed you it also correlates with the number of health clubs, and you can make a lot of correlations. And we need to try to extract from all these observations what might be causal. But what Bailey Hamilton did that I think is very important is she identified a group of chemicals, one of them being diethylstilbestrol, that when given at high doses causes weight loss, but at low doses causes weight gain. And that's the so-called non-monotonic dose response, which you heard mentioned earlier. And it's highly characteristic of endocrine disrupting chemicals and is completely an anathema to all toxicologists, because toxicology must be linear. Important point you can't see too well here is that many of these chemicals have an effect on the endocrine system. And as you know, I don't need to say to this audience, appetite and satiety and metabolism are under hormonal control in the body, and perhaps one of the most important of these is leptin. And the basal metabolic rate is set by the thyroid hormone receptor, which is a member of the family of receptors that I work on, the nuclear hormone receptor family. The development of fat cells themselves is also under hormonal control. And one of the key players is this gene with an absolutely terrible name, peroxisome proliferator activated receptor, gamma. And the name is an artifact of history, how it was discovered. But this family of genes is highly involved in lipid metabolism, and PPR gamma is considered to be the master regulator of fat cell development. If you express it in a stem cell, that stem cell is now a preadipocyte. And if you activate PPR gamma in a preadipocyte, the cell will differentiate to become an adipocyte. And if you activate it in an adipocyte, that cell will store more triglycerides. So it's a very important player in fat cell development and function. And we'll talk more about this gene later. So you also heard Monica define endocrine disrupting chemicals. And I think I follow a little bit different definition. The WHO and UNEP definition that you heard before was a kind of a committee decision, and it was influenced by industry. So the Endocrine Society of the United States came out with a slightly different definition, which is that an endocrine disruptor is an exogenous chemical or a mixture that interferes with any aspect of hormone action. And the key difference is that the WHO definition has that causes adverse consequences. And that is a huge point of controversy because industry says, well, if you can't prove adverse consequences, then it's not an endocrine disruptor, so prove it, go ahead. So as you can imagine, that's very difficult to do. So endocrine disruptors can be the wrong signal, the loss of the signal being expressed at the wrong place at the wrong time, and a whole host of other ways. One of the key points is that because hormones work at low concentrations, many of them parts per billion, so do endocrine disruptors. So how are we exposed? You heard a lot about persistent organic pollutants. You heard a lot about pesticides, personal care products. There are many routes of exposure which bring endocrine disruptors into our bodies. Here's just a partial list. You've seen some of these before. You heard about 2,4-D. Everyone has heard about DDT. And perhaps it's not so surprising that herbicides and fungicides and insecticides are also endocrine disruptors because these chemicals are toxic to life. They're designed to be toxic to life. But it's a big surprise that these industrial chemicals should also be endocrine disruptors. It's not intuitive that the water bottle or the beverage bottle that you use should be supplying hormonally active chemicals into your body. These fall into a whole host of categories. This summarizes them. We've by no means systematically examined all chemicals in use for their endocrine disruptor ability, but there's at least 1,000 of them. This is a bit of a silly picture, but it shows you another important route of exposure to endocrine disruptors, and that is we willingly put all kinds of personal care products on our bodies, many of which have endocrine disruptors in them. Phthalates are in there, parabens, there's a whole host of them. And if you doubt that these chemicals get into your body, I suggest you to read this book. It also has a silly name, Slow Death by Rubber Duck. But what these guys did is they measured baseline levels of chemicals in their blood, and they sat in a room, they sprayed an air freshener and sat in the room for 2 days and watched television and measured the levels of phthalates in their bodies. And they did this with a whole variety of chemicals and could show that just by living in proximity to these chemicals in common use, you will get them into your body at levels which are known to cause damage in animals. Well, the industry doesn't agree with this. I love this cartoon where it says, "We in the business community prefer a cautious wait-and-see approach over needless media scaremongering as the guy has grown breasts." And this is indeed the attitude of industry, that, well, there's no such thing as endocrine disruptors, or, well, if there are, you're not really exposed very much. And if you're exposed, well, the consequences really aren't so bad. Prove to me adverse consequences. And that's a point of view. I would counter with, well, then explain this increase over the last 50 years in reproductive diseases and disorders, the decrease in reproductive function worldwide over the same period of time. So perhaps it's a coincidence. Or perhaps there's a causal relationship between putting hormonally active chemicals into the environment and hormone-related disorders. So you need to make your own conclusion about that, but I think the data are fairly strong. So what I'd like to talk to you about are whether endocrine-disrupting chemicals might be involved in the development of fat cells and obesity. And this is a cartoon, and unfortunately this is reality. We see lots of very fat kids in America. And there was once a time when we thought, oh, the baby's growing really well. He's 99th percentile. You're doing a good job. And when my daughter was little, she was always around the 5th percentile, and her pediatrician used to say, you need to get her to gain more weight. We don't do that anymore, happily. And the problem is that kids that look like this often end up as adults that look like this. And there's a proliferation of these little carts in the United States. And it's sad that people have become so obese that they can't walk themselves around. And anything we can do to reduce the prevalence of this would be very welcome. So here are the players in our drama, the nuclear hormone receptors. In humans, these are a class of 48 different genes. And according to the Environmental Protection Agency, the genes of interest are the androgen receptor, the estrogen receptors, and the thyroid hormone receptors. So that's coded in law in the United States in the Food Quality Protection Act. I'm going to tell you about 2 other receptors that are targets for endocrine disruptors. And I think the point I'd like to make with this slide is there's no reason in principle to exclude any of these family of receptors from being targets for endocrine disruptors. We just haven't dug very deeply yet. And in principle, any of these could be targets. So we came up with, some years ago, based on the data, which I'll show you briefly, the idea that there could be such things as obesogens. And according to me, obesogens are chemicals that inappropriately stimulate the development of fat or the storage of fat, either by doing something directly to the fat cells or by altering the control of appetite and satiety. Now, is this my fantasy, as I was told when I first tried to write a grant to study this, or is there actually some data to support this idea? Well, it turns out that Retha Newbold and her colleagues at NIEHS in the United States showed that if you exposed mice for the first 5 days after birth to 5 parts per billion of diethylstilbestrol, you could get a mouse that looked like this, a very, very fat mouse. And Retha says these mice don't eat any more or exercise any less than the animals that are not exposed. And she showed that genistein has similar effects, and Fred Vom Saal and Beverly Rubin have shown that bisphenol A has similar effects. We know from human studies that there are drugs that make people fat. Thiazolidine-dione anti-diabetic drugs make people fat. They increase fat storage, the number of fat cells, and yes, that can have some beneficial effects, but the bottom line is that they make people fat. We know from epi studies, from Rick Stahlhut and other people, that urinary phthalate levels correlated with waist diameter and insulin resistance in humans. And you heard from Lars that many chemicals are linked with obesity and diabetes in epidemiological studies. We know from studies in cell culture that a whole host of chemicals can cause either pre-adipocytes or stem cells to differentiate into fat cells. And the ones that we work on, which I'll show you in a moment, are organotins, but there are many, many such chemicals. So I think that the existence of such things as obesogens is plausible, and it's not just my fantasy. So here's our favorite endocrine disruptor, tributyl-tin. And tributyl-tin is known, until recently, was one of the first and only endocrine disruptors for which there was no controversy. Tributyl-tin causes imposex, the imposition of male sexual characteristics onto females at parts per billion levels. You find it at parts per billion levels, so the cause and effect was really quite clear. So here's an example, and you can see that the normal snail has a very small penis, and a tributyl-tin exposed animal has an absolutely colossal penis. And as you can imagine, the parts don't fit anymore, and these animals are sterile. So I confess I'm not so interested in snails. I'm a vertebrate developmental biologist, and I was sitting very sleepily in the south of Japan in a meeting that was all in Japanese, and Professor Tanabe gave one of the few English talks, and he said that tributyl-tin could sex reverse genetically female flounder and zebrafish. And I thought, whoa, okay. That got my attention. And I called back to the lab and I said, guys, test tributyl-tin, not just on the one receptor we're working on, but test it on all of them. And we expected that it would have an effect on a steroid receptor. And what we found instead was the tributyl-tin bound to and activated at parts per billion levels, RXR and PPR gamma. And once we had that result, there was only one place to go. We had to study the development of fat. And it so happens that RXR and PPR gamma function as a heterodimer. So they work together as partners, and you can activate either half of that dimer and get the same effect. We found that it could induce adipogenesis in cell culture models at the same levels, parts per billion, and that prenatal exposure made mice gain weight. And that's published a long time ago, so I won't go into the details, except to show you one figure from the paper. And this is the testicular fat pad from either control mice or TBT-treated mice, and you can see that there's about a 15% increase in the size of this fat pad in 10-week-old animals. They ate a normal diet. These weren't high-fat exposed or anything else. They just ate the normal diet. But the mice weren't any larger. So the fat depot size increased at the expense of overall body mass. So we've been trying to understand, how did this happen? So the conventional wisdom would be that, well, we've changed the appetite of these mice, so they eat more. Well, they don't. Perhaps we've altered their ability to process and store lipids. We've made extra efficient fat cells. Or perhaps we've made more fat cells or more pre-fat cells, and that's the model that we favor. And the reason we favor that model is because it was already known that mesenchymal stem cells, or otherwise known as multipotent stromal cells, which can produce many lineages in the body, including bone, cartilage, and fat, can be induced to differentiate into fat cells when you treat them with rosiglitazone, Vandia, a PPR gamma activator. So that led us to believe that perhaps what's going on is TBT is changing this population of cells and making more fat cells out of them. So here's the mesenchymal stem cell, and you can see it's multipotent. It can go down a variety of pathways. And here's muscle, and here's cartilage, and here's bone, and here's fat. And what I'd like to point out is sitting at the bottom of this pathway, The switch, as it were, between the fat and bone lineage is PPR gamma. If you express PPR gamma, it goes down the fat lineage, and if you inhibit its expression, it goes down the bone lineage. So the experiment we did is we gave mice tributyl-10 prenatally, or we gave them rosiglitazone, or we gave them vehicle control, and we asked the question, what did we do to this population of cells, if anything? So I'll just summarize this data. What we could show is that in animals that were exposed prenatally to rosiglitazone or to tributyl-10, this population of cells made more fat cells. And that was reflected by the expression of fat-specific markers like fatty acid binding protein 4 and leptin. And when we asked the cells to differentiate into bone cells, when we treated them with a bone-inducing cocktail, what happened was the rosiglitazone-treated or the tributyl-10-treated animals didn't make bone cells. And they didn't express bone markers like osteopontin or osteocalcin, alkaline phosphatase. But despite that we told them to become bone cells, they became fat cells instead and expressed fat markers like fatty acid binding protein and lipoprotein lipase and a whole host of other fat marker genes. So we've changed the fate of these cells. So how does this happen? So this is a review from Peter Tontonos and Bruce Spiegelman. And basically what it says is that we know a lot about how cells get from pre-adipocyte to adipocyte. Here's PPR gamma. It integrates signaling from a variety of pathways, causes the expression of a whole host of fat-specific genes, and you make a fat cell. What we don't know is how do you get from over here to pre-adipocyte very well. We know a little bit. So we started surveying the genes in this column that are PPR gamma targets and asked the question, which ones of these genes had their expression changed in the cells from treated animals? And I won't go into detail here except to say that a number of them, the fat-specific genes, were upregulated, which suggested to us that perhaps we've somehow modified the regulatory regions of these genes such that they're expressed at higher levels. So this shows one bit of such data. So here we're interrogating the promoter of these genes for whether they're methylated or not. So if they're methylated, they're not expressed. If they're undermethylated, they are expressed. So you can see in this gene, 3 sites on this promoter are less methylated, which is associated with less expression. Having said that, we weren't so successful with all of the other genes, which led us to a different approach for this that I'll talk about in a moment. But this says perhaps what we're seeing here is an epigenetic change in the regulation of the stem cell fate that could be heritable. So we asked that question next, and in order to do that, we changed the approach. So in order to affect the next generation, you need to affect these animals early in gestation, and we've been giving a single dose of tributyl-10 later in life. So we changed to a drinking water dosing. And the other reason we did this was because the Environmental Protection Agency in the US couldn't care less about any other dose than oral dosing. So, okay, that's the way we'll do it. And we treated them with vehicle control or rosiglitazone or 3 different doses of tributyl-10 in the drinking water. And this middle dose here is calibrated to give the same dose over gestation as the single shot we were giving before. And the other important thing about this slide is this thing called the NOEL, which means no observed adverse effect level. And that's important to all of us because that is the dose that regulatory agencies use to set the allowable human exposure. So what they do is someone establishes the NOEL, usually the company that sells the stuff, and the EPA says, thank you very much, and they apply a safety factor of 100, and that's the human tolerable daily intake. So here you can see we're working largely below the NOEL. So we exposed females throughout gestation, and took the F1s, and we killed some, and we mated some, and went all the way out to F3. And we looked at a variety of parameters, body weight, fat tissue weight, the size, gene expression profile, and we also looked at the livers. If you see an effect in the first 2 generations, we call that a multi-generational effect. And the reason we distinguish is because these 2 generations were both exposed. So the F1s were exposed as embryos inside mom, and the F2s were exposed as germ cells inside the F1. If you see an effect in F3, that's called a transgenerational effect, and this is a permanent effect. These animals were never exposed. And Mike Skinner has shown that effects that you see in F3, he can also detect out at F6. So what did we see? So we didn't see much effect on body weight except with rosiglitazone, and we could see that a few fat pads were bigger in the F1 animals. These are young animals, 8 weeks old. If you look in F2, you can see effects on body weight and TBT, and all the fat depots are significantly larger. And you see the same in F3. What about the size of the cells? So surprisingly, actually, even though the fat depots weren't so much bigger in F1, the fat cells were very much bigger. And that persists in F2 and also in F3. What about gene expression in the mesenchymal stem cells? Have we changed that? So this is an interesting gene, zinc finger protein 423. It regulates the fat program. And in particular, it regulates PPR gamma, which is up in all three. PPR gamma regulates fatty acid binding protein 4 and lipoprotein lipase, which is up in all three. And look at the F3 generation. These guys are off the chart in F3. So this effect gets stronger in F3, which is the opposite of what you would expect if it were a genetic change. You would expect a genetic change to be diluted as you breed the animals. The epigenetic change is stable. And here's another gene, Pref1, that's an inhibitor of adipogenesis. So remember I said we didn't have much luck messing around with the promoters of these genes, and I met up with a guy named Toshi Shioda from Mass General at the Gordon conference a couple years ago. And Toshi said, you're doing this all wrong. We should just do deep sequencing and look in an unbiased way at the genome of these animals. So I sent him our last few dregs of material, and Toshi did deep sequencing, methylated DNA immunoprecipitation of these. You got a number of methylation peaks, and you could show that there were a bunch of peaks that were stronger in vehicle versus TBT, which means TBT caused less methylation, presumably more expression, and a bunch that were stronger, which means that TBT caused these genes to be expressed less. And I won't go into that, except to say that this shows, at least in a preliminary way, that we can detect epigenetic changes in the DNA from F3 animals that came from F0 exposure. And this was enough to get us a big grant from NIH, and perhaps the next time I come here, I'll be able to tell you what genes are involved in this effect. The other thing we saw is my postdoc, Raquel Chamorro-Garcia, said to me, you know, Bruce, the livers are white. What does that mean? And I laughed and said, that means you get to cut them, of course. So she did that. She sectioned the livers, and she stained them here for lipids in red. And you can see that the F1, the TBT animals, have a lot of lipid. The F2 have it, and the F3 still have it. These are males. Interestingly, females are the same except with rosiglitazone, which females show an effect in F1. Now, these animals were not challenged with high-fat diet. These animals were living on a normal diet, and they just accumulated fat in their liver as a result of this prenatal exposure. And that's also reflected in gene expression. This is lipid synthetic pathway, PPR gamma, SRABP1, fatty acid synthase, and the message is these are all upregulated. Fatty acid oxidation is also upregulated, so the liver's trying to compensate in these animals, and you can see that in all three generations, fatty acid oxidation is increased. And this gene, FSP27, coats vesicles, so it's required for lipid storage, and you can see it's also up. So what have I showed you? I think that organotins are very potent activators of these two nuclear receptors, RXR and PPR gamma, and they do it at parts per billion levels, at the kind of levels that we can see in the environment. It drives fat cell differentiation in cell culture. Very importantly, in vivo exposure during development causes later adipogenesis in two species that we've looked at, mice and frogs. Other people have looked at rats and a variety of fish. It also inhibits bone formation in culture, although I didn't show you those data. The effects of this maternal exposure are transgenerational. That is, they're heritable at least out to F3, and in our current study, we're going to go out to F4. There's multiple potential modes of action. I've shown you data for this mode. There's no reason to exclude the possibility that other pathways might be involved, but we simply haven't demonstrated that yet. So people always say, well, is tributyltin making me fat? And the answer is, well, we know that adult exposure turns on the fat program. Importantly, we know that drugs that activate PPR gamma make people fat. So why wouldn't chemicals that activate PPR gamma have the same effect? The big question is whether humans are exposed. And the answer is, as you also heard, we don't know. There's only a very few studies showing whether humans are exposed to TBT, and those studies show that the levels are in the range that we activate the receptors. PVC plastic has quite a bit of organotins in it, although the plastic industry is very happy to say we don't ever put tributyl tin into our plastics, and they don't. What they use is dibutyl tin, which is contaminated with tributyl tin, and they call that a non-intentionally added substance because they didn't mean to put it there, but it's still there. So is the environment making us fat? I'm not sure that the data would support that statement, but I think that what the existence of obesogens shows us is that there are other factors than the traditional risk factors that we've thought about that perhaps we ought to consider in trying to understand this epidemic of obesity. So how many obesogens are there? I've shown you data for organotins. There's pretty good data for phthalates and perfluorinated chemicals. There's a good, solid data set on estrogens. We know a fair amount about nicotine, air pollution. Who's heard Rob Lustig shouting from the rooftops about fructose? Fructose is a very potent obesogen. We've shown that fungicides are also potent obesogens. There's a lot that we don't know, the rest of the iceberg. So is this it? Are there 20 obesogens or 200 or 2,000? Wrap it up. And the answer is we don't know. From our preliminary studies, I would say there's probably going to be hundreds. We know almost nothing about the body burdens in the population. We would like to know that. We don't know what are the targets of action or the critical windows of exposure. I've looked at developmental exposure because that's my interest, but what about other windows? We don't know much about how this prenatal exposure alters the adult phenotype. We think that it's epigenetic, but we need to learn a lot about the mechanisms involved. What are the implications for human health? I think the most important implication is that calories in, calories out, all calories are the same, is not the explanation for the obesity epidemic. And especially we know that because Rob Lustig has an epidemic of obese 6-month-old babies. And I think it's pretty clear that 6-month-old babies do not eat more and exercise less than they used to. Babies eat until they're full and they exercise when they're awake, and that's the way it is. We know that there are such things as obesogens that inappropriately stimulate fat development and storage because there are pharmaceutical obesogens. There are drugs that make people fat. Thiazolidine diones make people fat. Atypical antipsychotics and tricyclic antidepressants make people fat. Why would you expect that chemicals that hit the same pathways not to have the same effect? That would be unreasonable. There's a whole host of environmental contaminants that can lead to obesity in animals and presumably in humans. We know that prenatal obesogen exposure reprograms exposed animals to be fat, and we think at least part of that has to do with epigenetic reprogramming of stem cell fate such that we make more fat cells. And here's a silly cartoon I found, how his epigenome has interacted with his diet to make him a blimp. Seriously, I think that the existence of obesogens says very strongly we have to really shift the paradigm from doing bariatric surgery on obese people to preventing kids from being obese in the first place. And we do that by reducing exposure to obesogens, by optimizing nutrition, by doing the best that we can. I'd like to just point out quickly that there are other chemicals besides tributyl tin with transgenerational effects. Mike Skinner has a whole host of papers showing that vinclozolin, hydrocarbons, plastic components, and also DDT cause transgenerational inheritance of obesity. Last, I'd like to acknowledge the people who did this work. Originally, the obesogen work in my lab was started by postdoc Felix Grun. Severine Kirchner did the first stem cell work. Raquel Chamorro Garcia is a postdoc who leads the transgenerational study. We've had funding from a variety of agencies that we appreciate very much. And our collaborators, Toshi Shoda and Tayaguchi and Jun Kano and Kotaro Azuma in Japan, who have all helped us to understand what's going on here. And I'll leave this slide up about human studies that support the obesogen hypothesis while I thank you for your attention and entertain the questions you might have. Thank you.
My first word I'll say again, thank you for the excellent lecture. I really enjoyed it. But I want to know if you think that there is any genetic predilection to be more sensitive, that when you are just born or before, you are more sensitive to environment than others, and then you will be fat, and other one won't be fat, even though you were born in the same place.
So The question was, do I think there's any genetic predilection to being more sensitive to obesogen exposure? And I would say that there very well should be, because we are all very different from each other. Some of us should be more sensitive and others less sensitive to the same exposure, whether it's a chemical exposure or whether it's environment, air pollution, anything you can name, there will be a variety of sensitivity to. Our animals are inbred, so they don't differ much in their sensitivity.
Do you have any data about that? Any genes that you know that make us more sensitive?
That make us more sensitive to chemical exposure? There's no data on that, not to my knowledge.
Next time you will tell us about it.
Yes, I hope. Okay, thank you very much.